So what we're going to do today is we're going to talk a bit about the new Ensembl release, which is Ensembl 76, and talk about some of the things that are new in this new release. So I've got all your microphones muted, but I can take questions at the end. If you want to type questions into the chat box as we're going along, please do, but I won't be able to pay attention to that while we're going through the presentation. I'll have a look at that when we get to the end. Maybe some of the questions will already have been answered. Maybe some of them have been new. There might be a slight lag in slide changing. There is a PDF. If you go into materials on your Go to Training control panel, you should be able to download a PDF, which is e76_webinar, and you can take that away and have a look at it at any time in the future if you like. So what I want to go over today, some of the new things that we have. We have a new genome for human. We've got some new and updated tools. So we've got a new version of BLAT. We've got new ways to export particularly sequence data, which are hopefully easier to use. We have a new regulation build, which I'll be talking about, and we also have a couple of new species. So the new human genome assembly is called GRCH38. It's also known as HG38. So if you will also see that in some publications and some databases. This was produced by the Genome Reference Consortium, and it came out in December. So that just gives you a bit of an idea of how much work it's actually been to annotate this new human genome, that it's now August that we've actually released it. This includes integration of what we call fixed patches into the genome. There are more haplotypes, and there's now centromeres in the new genome assembly. One of the sort of downstream effects of this is we have a completely new coordinate system for the genome. So any coordinates you have, any numbers, they will change. It also means from our perspective at Ensembl, we had to remap everything, hence the 7 months of work involved. We've had to integrate fixed patches. So in the old genome, in GRCH37, also known as HG19, there were gaps in the human genome. The way that we found around this was to produce what were called patches. These would cover up the gap, and that meant that genes which were incomplete could then be completed on the patch. In the new genome, we've gotten rid of the old gap, and we've just integrated the patch in. The patch is now what is the genome. So that's a real leap forward, a really good thing that's happened because of the new genome. We also have more haplotypes. So these are versions of the genome, really sort of highly variable regions where individuals are known to have maybe a different set of genes or genes in a different order. The most well-known of these is the MHC region on chromosome 6, which contains the minor histocompatibility complex. We have got a lot more haplotypes in GRCH38. A lot more of these regions have been identified with a lot more sequencing has been done, which means that we found a lot more of these at different points in the genome. If you look at our different views, if you look at this chromosome view, you can see that we have a lot more of these red haplotypes in E76 compared with E75, but we've lost all of these greens, and the greens are the patches. So what does this mean for the average user? The first thing, it means that the coordinate systems all change. So any gene that you'll be looking at will have a new set of coordinates. However, within those coordinates, the models mostly have not changed. If they have changed, there's probably a reason for it. It's quite often because there was something wrong with it before, and the new assembly has fixed what was wrong there, and that's why the model has changed. If you look at something like variation data, you might see that the variant ID will be the same, but the coordinates have changed. And in some cases, you'll see that the reference and alternative alleles have flipped over because the new assembly contains the major allele as the reference and the minor allele as the alternative. And in some cases, in the old assembly, the reference allele was the minor one. If you don't want to change, if you've still got analyses going, if you're still working with GRCH37, we do have a dedicated site, which is trch37.ensemble.org. And if you're using our REST server, we also have a special REST server for that, which is grch37.rest.ensemble.org. We will be updating this site periodically to make sure that it gets all of the software updates and things that you'll see in the main Ensembl site, but we won't be updating it as often as you will the main site, and we won't be updating the gene annotation. If you're particularly interested in what goes on in a new genome assembly, we do have another webinar coming up in September, and this is involving the GRC, so the Genome Reference Consortium, who made the genome. So that's Kirsten Howe, who will be coming to help with that. We'll also be including Bronwyn Aiken, who is in charge of Ensembl primary analysis, so that's things like gene annotation, Carlos, who was involved in annotating the new genome, and also Julieta Spudic, who's from the outreach team. So there's a link there. If you go to the slides that you can download, you can click on that link and find out more about registering for that webinar. We have a new BLAST. That's a new thing that's come out in Ensembl 76. So we have a new input form. Hopefully this is easier to use. It allows you to submit multiple queries with titles, and it's hopefully a much nicer interface to work with. Also, if you're altering the parameters using our new input form, these are much easier to understand. They're written in human readable form rather than in things like letters that you then have to Google to find out what they mean. So hopefully that will make things a lot easier. The output format has changed as well. Hopefully, again, this will be a lot clearer to use. A lot of the links that you can go to are much nicer and easier to look at. And we have a table of jobs. So as you run BLAST jobs in Ensembl, you will now get a table showing you all the jobs that you've run, so you can go back to your previous queries much more easily. You've always had ticket numbers to get to your old tickets, but this allows you to do it without saving ticket numbers. You can save these to your account, and you can go back to your tickets. You can see how they're getting on. You also have, it automatically refreshes to tell you whether or not your ticket can be retrieved, rather than the old system where you had to keep clicking on retrieve. So it's much nicer to use. We have an easier way of doing sequence export. So previously, when you wanted to download a sequence from Sequence View, there was a blue button at the side that you had to click on for exporting the sequence, and there was a different button for exporting as rich text format. So if you export the sequence, you just get plain FASTA. If you want to get the annotation, all the colours, and all the markings that you've put on the sequence, then you would export as rich text format. We now have a single button at the top of the sequence, which will hopefully be more easy to find, which is download sequence. And that will let you choose whether you want the FASTA or the rich text. There's also a button alongside it for BLAST this sequence, so you can plunge your sequence straight into BLAST without having to select and copy and paste, which can make things a lot easier as well. And we also have our new regulatory build. So previously we had our build look like this, and now it looks like this. So it has changed a little bit. We've got different colours. Our blocks look a little bit different. And I'm going to explain a bit about how this build works, and hopefully that will explain what all the colours and what all the blocks mean. So we start with raw data from various projects. At the moment we have data from ENCODE, Roadmap Epigenomics. We're hoping to integrate in data from a number of other sources. So we're working with Blueprint, with HipSki, and we're keen to see if there are anything else out there that we would like to integrate in. So we take raw chipset data from these projects, and we use exactly the same process with all of these different, all of the data from these different projects. So for each cell line, we process the data to find patterns of activity, generally seen in particular features. So we look to see the kind of data we're looking at are things like transcription factor binding, like histone modification. So we'll look to see whether there's a cluster of histone modifications that we always see together. And when we see them in promoters. So then we can say in other places where we see these, We also think that they're promoters. So this gives what we call segmentation for each cell line. We then combine the segments for each cell line and say for the genome as a whole, what kind of features are there? Are there promoters? Are there enhancers? Are there insulators? And then for each feature in each cell line, we call the activity. We say, is it on or is it off? So let's have a look at how that works. We have cell lines which have transcription factor binding data. We call peaks for each of these. We use complicated algorithms for these, so there's smoothing that goes on, there's normalization that goes on against the baseline to produce these peaks. We then say in our two different cell lines, what's clustering together and what do we generally associate those features with? And this is what we call the segmentation. So we'll call particular regions in this cell line 1. We have some features and we've called it as this particular activity. In cell line 2, we haven't called the same activity. Whereas this region, we've called one kind of activity and we've called the same thing in the other cell line. From this, we can say overall, in the genome as a whole, what activity is going on where. So in our multi-cell track, we can say that this block here, there's a region here across all our cell lines which has this yellow activity. There's a region here across all our cell lines that has this red activity, and there's a region here that has this turquoise activity. Note that we don't have to see the activity in all the different cell lines. We're just saying that that activity has been seen overall. We then call each of these blocks. So these multi-cell blocks are made using 200 base per bin. So they're asking what the activity is in 200 base per bin. So they are fixed size. We then say for these fixed size blocks, what's the activity in each cell line? So in cell line 1, we can see that this yellow region is active. So we colour the block in active. In cell line 2, we see that it's not active. So we colour it in grey. This red one is active in both, and this turquoise one is active in cell line 2, but inactive in cell line 3. So that's how we produce our build, and then in real life it actually looks very much like this, but a little bit more detailed and a little bit more messy. So this is what it looks like in our browser. We have our multi-cell features, so we have our features, and then I've got two cell lines showing here. I've got GM12878 and HeLa cells, and you can see that this region is active in both, which is a promoter. We've got a couple of CTCF binding sites here, which are inactive in both. And we can see that we've got peaks here. So this is our raw chipset data. And we can see that we've got peaks of CTCF binding here and here in both of the cell lines, which corresponds to the block. So you can see how the data was, how this raw data was used to produce these peaks. Now you can view these in our region in detail page, and the way that you find them hasn't changed. But if you go into the features themselves, if you click on them and go into what we call the regulatory tab, you end up with these easy to use menus which allow you to turn on features for your different cell types of interest. If you're interested in further information about our regulatory build, we've got some blog posts. So there's a post on what the build is, and there's a post on how we made it. These were both written by Daniel Zabino, who leads our regulation team. There's also a blog that I didn't have time to write about, which came out earlier today, which tells you how you can get this, our regulatory build as a track hub. And we're also going to have a video coming soon. I'll be working on it tomorrow. That will go onto our YouTube channel about how to use the new data. So it'll describe in detail how all these menus work and how you can get to them. We've got a couple of new species. The first one is the Amazon molly, which is a fish. She's not called the Amazon molly because she's from the Amazon. She's called the Amazon molly because they're all female. They have asexual reproduction, which makes them very interesting animals. And we've produced a gene build using RNA-seq, but also using homologous fish genes. And she's of interest because she has this asexual reproduction. So people are interested to find out why the evolutionary benefits or costs of sexual versus asexual reproduction. And she's also used to study carcinogenesis because she's very easy to keep in the lab. There's a blog written by Daniel Murphy, who's worked on the gene build for Amazon molly, all about how the Amazon molly is interesting as a species. The other species we've got is the olive baboon, which joins our Ensembl primates group. This was a standard Ensembl gene build, but there is tissue-specific RNA-seq data available. And we're interested in the olive baboon. It's used to study psychology and behaviour, and also for comparison to other primates that are available in Ensembl, such as, of course, human. A few other things that have come out. We've updated our gene builds for human and mouse. So for human, we now have gen code release 20, and for mouse, gen code release M3. The genes that you'll see in Ensembl that are produced by Ensembl are the gen code genes for human and mouse, and so these have been updated. We also have updated variants. So we've updated to dbSNP 140 for some farm animals, chicken, cow, pig, and sheep. For human, we've updated to Cosmic version 69. So Cosmic's a database of somatic variants found in cancer. We've also got a lot more structural variant data from DGVA for a number of species. And we've also got some more human phenotype data from both ClinVar and Decipher. There is absolutely loads of help and documentation available. We've got online courses available on EBI Train Online, as well as a tutorials page on Ensembl. We've got a YouTube channel. I mentioned this before. We will be putting our new regulation video on the YouTube channel, and we also have a UQ channel if anybody's in China at any point and would like to get access. If you do have any questions about anything that you've seen in this webinar or anywhere you see on Ensembl, do send us an email to helpdesk@ensembl.org. Sorry, going back to the YouTube channel, I will be putting a copy of this webinar on the YouTube channel as well. So if you have a colleague who missed the webinar who would like to have come, or you'd like to look back at it in future, then we'll make that available there. Do follow us on our social media, on our Facebook, our Twitter, and our blog. I've pointed you to quite a few blog posts here, so hopefully some of those will be of interest. We also have a lot of publications. If you do use our data, please cite us and have a look at our most recent. We put out a nucleic acids research paper every year, which just details what we've been up to for the past year. So if you want to know what we've been doing, do have a look at that. These are the people who work on the Ensembl project. So all of this work, the 7 months of producing this new human genome, was done by this very large team here. So you can see how much work it might have been. And just a few names and a few funders as well, who put their money into the project and allow it to exist. So we are very grateful to them. So that's the presentation for today. Now will be time to ask some questions. So if you type your question into the chat box in the training interface, I will answer it. You can download the materials from here. You'll also get an email which has got them attached as well. So if you don't manage to download those, you will get it via email. And there's a quick feedback poll when you log out as well. Okay, so Richard asks about patches in the new human genome. So at present, there are no patches, but we are already aware of regions that need them. There will be patches coming out. We don't know when. It all depends on when GRC release them. So patches, Richard says, will that affect coordinates? So patches don't affect coordinates. That's kind of the idea behind them. So when we produce patches, we're putting the sequence on top of the genome rather than integrating it in, which means you don't have to produce new coordinates. This is why a new assembly is so much work, because it changes the coordinates. Patches make things slightly more confusing to use, but they allow continuity because they don't affect the coordinates. So do we annotate following the application of a patch? We annotate the patch. We don't annotate around the patch. We don't annotate anywhere else. So the patch will be fully annotated, probably with the improved gene structures and things that are on it, but nothing else will be affected. We do pull in new genes, so any new data will be further annotated on top of what we already have, but we won't change what we already have. Just if there's a patch, only what's on the patch. So Chris asks if the archive server will be as powerful. It is missing some features, but I think there are a few things you can't get at. Off the top of my head, I can't think what they are. We're trying to keep it as up to date as we can. We've always kept archives. So one of the things that we haven't kept in archives is things like BLAST. For GRC H37, we decided to keep the BLAST in our archive. So it is better than the archives. It's also, as with everything, it's accessible via our Perl API. It's got its own REST server, so it's accessible via that. So there's a lot still available. I can't think what actually we have lost. I'm sure somebody told me there was something. If there are no further questions, I'll close the training now. You'll receive an email in about an hour, which will have the slides on it. Thank you very much for attending. I hope this was useful to you.